Welcome to Test Support Center, your source for any ACT, SAT, AP, or MCAT assistance. Today, we will be discussing biology, and in more specificity, nucleotides and nucleic acids. Biology is a very complicated subject. The cell is unbelievably complex, and in order for us to understand the complexities of the human body, we must first understand the inner workings of the most basic building block in all biological systems, the cell. In our journey of understanding, we will begin with the smallest unit of information found in these cells, deoxyribonucleic acid, or DNA. DNA and RNA are nucleic acids, or acids that are formed in the nucleus of the cell. DNA, as stated before, is an acronym for deoxyribonucleic acid. Similarly, RNA is an acronym for ribonucleic acid. The building block for DNA in the cell are things called deoxyribonucleoside 5-triphosphates, or DNTPs. These deoxyribonucleotides are built from three primary structures. The first being a monosaccharide or simple sugar called ribose. Ribose is seen right here on the whiteboard, and it's important to understand the nomenclature going forward. What we actually do is label the carbons of ribose as such. This carbon being carbon 1, this carbon being carbon 2, this carbon being carbon 3, this carbon being carbon 4, and this carbon attached actually to carbon 4 is carbon 5. The second component is one of the 4 aromatic nitrogenous bases. Aromatic simply means that these molecules contain an aromatic ring. Nitrogenous also means that these molecules contain a nitrogen atom, and base means that these are basic molecules overall. These are all very simple chemistry terms that you are already probably very familiar with. The list of the 4 bases that are found in DNA are as follows: guanine, adenine, cytosine, and thymine. Uracil is another base that is only found in RNA and is something that we will discuss in a later video. How we differentiate between these sets of bases is on what they are actually derived from. So guanine and adenine are derived from a molecule called purine, which is why we call them the purines. Cytosine, uracil, and thymine are actually derived from a molecule called pyrimidine, which is why we call these the pyrimidines. This also leads us to a very easy acronym, or mnemonic actually, which helps us remember this side of this column. Pyramids, which have sharp edges, cut, which are the first 3 letters of the 3 pyrimidines, cytosine, uracil, and thymine. Very important thing to remember about these purines and pyrimidines is that they have high hydrogen bonding potential. A nucleoside with an S is a ribose attached to a purine or a pyrimidine. In this example, it is guanine, at the 1 carbon via a beta nitrogen glycosidic linkage. Remember from your organic chemistry days that a beta tag on a bond simply means that this carbon-nitrogen bond is cis, or on the same side as this hydroxyl group that is attached to the 5 carbon. Each nucleoside is differentiated based on what base is actually attached to the ribose. So the way they name these is based off of if there's a guanine, adenine, cytosine, or thymine, or a uracil attached to one of these riboses. So in the case of an A ribose, we call it adenosine. A G ribose is called a guanosine, a C ribose a cytidine, a T ribose called a thymidine, and a U ribose called a uridine. The final component of these DNTPs is a phosphate group. Nucleotides with a T are phosphorylated nucleosides that can contain up to 3 phosphate groups at the 5-hydroxyl group. When said nucleotides contain 3 phosphate groups, they are referred to as deoxynucleoside triphosphates, abbreviated as DNTPs. The ubiquitous energy molecule, ATP, is actually a nucleotide. This is something that you've probably heard of over and over again in your AP biology classes, or even honors biology classes. But the energy currency in all of our cells is actually a simple nucleotide. The ribose plus phosphate portion of a nucleotide is referred to as the backbone of the DNA because it doesn't change. It's not a variable. Every nucleotide is going to have a phosphorylated ribose. The nitrogenous bases is what actually varies in between nucleotides. Therefore, the 4 different DNTPs only differ between the 4 different bases: guanine, adenine, cytosine, and thymine. Nucleotides in DNA are linked together via phosphodiester bonds between the 3-hydroxy group of one nucleotide and the 5-phosphate group of another nucleotide. The end of the chain with the free 5-phosphate group is generally written first in a polynucleotide sequence. Subsequently, the nucleotide with the free 3-hydroxyl group is written last. Since the only thing that varies in these nucleotide sequences are the bases that are attached to the 1 carbon on the nucleotides, we generally can abbreviate these sequences just as the first letter of the base that is attached. So in this case, we have a guanine and a thymine attached to our nucleotide sequence, which means that we would actually abbreviate this as GT. So a quick checkpoint question for all of you is this one right here. I would recommend that you would pause the video right now and create your own answer before I explain mine. So the question is, which nucleotide in the following oligonucleotide sequence CAAT has a free 5-phosphate group? Remember what we said earlier, that the first one written is generally the one with the free 5-phosphate group, meaning that in this case, the nucleotide with the free phosphate group would be the one containing a cytosine. Lastly, we will discuss the Watson and Crick model of DNA structure. James Watson and Francis Crick were 2 biologists that developed a model for the structure of DNA. They received a tremendous amount of assistance from Rosalind Franklin, a name that often gets forgotten in history. Watson and Crick's model is, simply put, a double-stranded right-handed helix that is held together by a hydrogen bond between the nitrogenous bases. The model is extremely important for us to understand. So we have been treating polynucleotides as long chains, but in the cell, they are not found in this form. Instead, DNA is found in the nucleus in a double-stranded fashion. This means that 2 long polynucleotide chains are hydrogen bonded together in an anti-parallel orientation. This means that the 5 end of one chain is hydrogen bonded to the free 3 end of another. This anti-parallel orientation may be reminiscent of the beta pleated sheets when describing secondary protein structure, which is something you might have learned in your biochemistry class. These hydrogen bonds between the bases are very, very specific. Adenine is always bonded to thymine, and guanine is always bonded to cytosine. This means that a purine is always bonded to a pyridine. The CG pair contains 3 hydrogen bonds, while the AT pair only contains 2. The bonding of 2 complementary strands of polynucleotide chains is called annealing or hybridization. On the contrary, the separation of these 2 chains is called melting or denaturation. The temperature at which a solution of DNA is 50% melted is termed T sub m. We have a quick checkpoint question for you. I would recommend that you pause the video and create an answer of your own before I explain mine. The question is, which T sub m would be higher? The T sub m for a chain ATT-GATC and its complement, or the T sub m for a chain CCG-TATGG and its complement. Remember what we stated earlier, that CG have 3 hydrogen bonds, while AT pairings only have 2 hydrogen bonds. That means the more CG bonds, the more hydrogen bonds, which also means that it would take more energy to break more bonds. Therefore, the T sub m would be higher with a chain that has more C and G bonding. So just looking at these chains, we would expect that the CCG-TATGG chain would have a much higher T sub m because it contains many more C's and G's. If this was a little bit harder to tell just by looking at them, you could also count out how many hydrogen bonds there are in them, which we'll do right now. So we can count right now. 2, 4, 6, 9, 11, 13, 11, 13, 16. So there's 16 right there. Hydrogen bonds. In this chain, we have 3, 6, 9, 11, 13, 15, 18, 21. So as we can clearly see, this chain has 21 hydrogen bonds, which means it would take much more energy to break 21 hydrogen bonds, making a higher T sub m than it would to break 16 hydrogen bonds. The last thing that we must understand about the Watson and Crick model is that not only is it double-stranded, but it is coiled. Instead of being like a ladder, like what we see here, it is a right-handed helix, meaning that it is turning in a clockwise motion. The bases are on the interior, while the backbone, remember, which is just the ribose plus the phosphate, is on the exterior. This entire helix is actually held together by not just hydrogen bonds, but van der Waals interactions and the hydrophobic effect, which are the other topics that we'll discuss in later videos. To wrap up the lesson, we have a few final questions. Remember, pause the video and create answers of your own before I explain mine. So the first question is if 15% of a double strand is cytosine, what percent of the DNA is adenine? So the best way to remember this is that every cytosine is going to have one guanine, and every adenine is going to have one thymine. That means as of now, the C's have 15%, and so do the G's. So overall, these equal 30% of the strand. That means if you take 100% of the strand and minus 30, you're going to get 70%. This is going to be split between the adenine and the thymine. So therefore, we just take this in half, and that's what percent of the DNA strand is adenine. That means adenine is 35%. And also, thymine is as well. So that's the first question. And then the last question is, are these anti-parallel? GCAATC and GATTGC. At first, you might be like, of course not. The G's don't match up, the CA doesn't match up, the ATs do match up, these don't match up, these don't match up. But you're forgetting. This is the 5 side. This is the 3 side. This is also the 5 side. And this is the 3 side. So really, we would have to flip this, or one of the two, and make sure the 5 and the 3's match up. So we'll do C, G, T, T, A, G. Now do these match up? GC, CG, AA, AT, or AT, AT, TA, CG. They do. So yes, these are anti-parallel. So that wraps up our video lesson on the biology of DNA. Like the video if it was helpful, subscribe if you would like to see more videos on this topic, and comment any questions or concerns you may have. Follow us on social media, all the links will be in the description, and thanks once again for watching. We'll see you later.